Hi, my name is Gilad Bino, and I'm a research fellow at the Center for Ecosystem Science at the University of New South Wales. First, apologies for not attending in person, and second, apologies for my voice, which just came back after a cold. I'd like to present some preliminary findings as part of the Platypus Conservation Initiative, which has a broad aim of developing strategies to keep platypus from extinction. And although the notion of an extinct platypus might sound far-fetched, humans have been driving many species, including some previously common species, to extinction, and unfortunately Australia thus far has had quite a horrible track record in terms of extinction, with the worst rate of mammal extinction in the world. The findings I'll be presenting has been developed and collected by myself and Tennille Hawk, a PhD student at the center, and Professor Richard Kingsford. As I'm not there in person, a proper introduction is due. Here's a picture of lovely water and myself, and here's another, perhaps less appealing one, with Tennille by my side. The platypus is an extraordinary creature, displaying both mammalian and reptilian characteristics. You'll notice how it moves more like a crocodile rather than most mammals. It's only one of 5 egg-laying mammals. Venomous, the platypus carries spurs on their hind legs, and is a unique global species and an Australian icon. In 2016, the IUCN Red Listing, which is a global standard for assessing the extinction risk of species, elevated the conservation status of the species to near threatened, which implies a decline not exceeding 30% in range and numbers over the course of 30 years. This was based on only a small number of long-term monitoring programs detecting declines, as well as the large number of threatening processes which we know impact the species and its habitat. Unfortunately, Australian legislation is lagging, with only South Australia listing the species as threatened. You might be surprised to know that the platypus is currently not listed under the Environment Protection and Biodiversity Conservation Act. We know that there are a large number of threatening processes impacting platypuses and their habitat, including pollution, river regulation and fragmentation by weirs and dams, destruction of riparian vegetation, and unrestricted access of livestock to rivers, both destroying riverbanks and increasing sedimentation, thereby reducing habitat availability. Invasive species likely increase risk of overland movement and perhaps lower reproduction success. Direct mortality by closed yappy traps or opera houses, as well as hair ties and loose strings, cause strangulation. Increased awareness of local declines and extinction of platypuses, particularly within scientific circles, was the motivation to develop the Platypus Conservation Initiative, which is a partnership of all relevant state and federal agencies, as well as 3 universities, University of New South Wales, Sydney, and University of Melbourne, as well as Taronga Zoo. The Platypus Conservation Initiative currently has 3 aims. First, to evaluate past and present distribution and to quantify changes in trends. Second, to quantify possible impacts of river regulation and fragmentation of populations by large dams. And third, to develop models that would allow us to assess extinction risk under different scenarios, including both management and environmental change, that can help us develop conservation strategies that will help plan for the future of the species. To evaluate past and present distribution, we collated records of platypus observations made over the past century, held in state and national atlas datasets. In addition, we looked through digitized historical newspaper records mentioning sightings of platypuses. Here, we plotted all such observations, extending all the way far north as well as western flowing rivers of the Murray-Darling Basin. By stratifying areas into small subcatchment units and recording in each one the number of years since the last platypus observation was made, we can identify areas where we suspect local extinctions or significant declines have occurred. In green are depicted areas with observations of platypuses made over the past 10 years. In yellow, between 11 and 20 years, and in red, areas where the species hasn't been observed for over 20 years. In gray, we can also highlight areas where we suspect platypuses may have or still do occur, but we have absolutely no record there, which reinforces the significant knowledge gaps we still have with regards to platypus distribution and must be addressed if we want to accurately assess the extent of decline. Most of the contraction in extent have likely occurred in the western flowing rivers of the Murray-Darling Basin, as well as some areas in the north. To assess potential impacts of river regulation and fragmentation of populations by large dams, we embarked on 3 years of field surveys, comparing populations above and below a large dam and comparing those to a nearby largely free-flowing river. In our first year, we surveyed populations in northern New South Wales on the Severn River, above and below Pindari Dam, and compared those to the populations on Tenterfield Creek. In our second year, we focused on the Snowy Mountains region, looking at platypus populations on the Yucambine River, above and below Yucambine Dam, as well as those on the Threadbow and on the Snowy River directly downstream of Jindabyne Dam down to Dalgety. In 2018, our third field season, we surveyed platypus populations above and below Dartmouth Dam on the Midamida River and compared those to platypus populations on the Ovens River. Over the course of 3 years, we handled over 200 platypuses, trying to collect as much information as we can. In the field, we'd be setting up mobile lab, anesthetizing the animals, and going through a thorough assessment and sample collection. We've been collecting measurements and body condition, blood and biopsy for assessing genetic diversity and relatedness, collecting ectoparasites, namely ticks, for disease loads, swab, feces, and urine for health, fur, nail, and macroinvertebrates stored in the animal's cheek pouch for understanding diet using stable isotope analysis, as well as marking individuals for assessing population sizes and demographic structure. Finally, to some results. Here we can see the average number of platypuses caught per night across each of the rivers we've surveyed over the past 3 years. On the left, in green, are the rivers of northern New South Wales, in the middle the Snowy Mountains in blue, and on the right, northern Victoria. Marked with a red asterisk are populations directly downstream of a large dam. What's apparent is the large variability in the number of platypuses we've caught. I'd like to first draw your attention to the right, the rivers of northern Victoria, where we generally recorded the lowest number of platypuses, perhaps a result of habitat destruction and quality, along with recorded impacts due to extreme droughts in the area. But also note the extremely low numbers in the section directly downstream of Dartmouth Dam, where we also recorded very low water quality conditions and macroinvertebrate abundance and diversity, food for the platypus. These are likely a result of water regulation and management by the dam. To just quickly jump to the left, note also the lower number of platypuses directly downstream of Pindari Dam on the Severn River in northern New South Wales. And finally, to the Snowies in the middle. On the Snowy River, downstream of Jindabyne Dam, we recorded relatively high numbers of platypuses, as well as on the Yucambine, which appear to offer good platypus habitat. However, on the Threadbow River, we encountered relatively low numbers, but we cannot at this stage offer further insights. A quick look at the demographic structure in terms of the ratio of adults in dark to juveniles in gray, we can again see the large variability across different rivers. Note that in some rivers we recorded very low proportion of juveniles. This included the Snowy and Yucambine rivers downstream of Jindabyne and Yucambine Dam. Looking at the timing of juvenile emergence on the Snowy River, as shown here as the number of juveniles caught each month, you can see that we captured juveniles over the months of February, March, and April, and none were captured later in the year between August and December. This is quite consistent with what we know about the breeding behavior, with some platypuses in the region starting to breed around August. From observations in captivity, we know that gestation period is between 2 to 4 weeks, followed by incubation for another week, and hatchling, or called puggles, are suckled between 120 to 140 days. Based on emergence of juveniles, this suggests that puggles may be in burrows as early as September through to November on the Snowy River.
We also investigated movement behavior using acoustic telemetry by tagging 8 platypuses and deploying listening stations along the Snowy River. Here we can see the locations of stations we deployed along the Snowy River between the dam and Dalgety over 2017. In this particular study, we looked at large-scale movements of platypuses over 6 months between January and June. In the bottom plot, you can see the number of detections by each of the stations in relation to distance from the dam.
Again, variability is the case when looking at the minimum daily linear range covered by each platypus, which you can see here generally extended under and around 1 kilometer, but with some males covering distances up to 4 kilometers daily. We can also note the juveniles at the top only covering a small ranges during their first months in the water. Now I use the term minimum as our estimates are derived from passive listening stations, which means we can only measure what we observe. At the scale of months, we can appreciate the longer distances covered by some individuals during their foraging or territorial behaviors extending over 2 kilometers per month for male platypuses. We can also see the large differences between males and females in terms of distances traveled. We also observed increasing greater distances covered by male platypuses from April onwards, perhaps in relation to the onset of the breeding season in August, or perhaps changes in availability of food. Towards August 2017, we set out to try and evaluate whether we would observe changes in either platypus numbers or movement behavior in response to the large spring release planned on the 4th of October, releasing over 8,000 megaliters with a peak flow rate of 13,000 megaliters per day. I was hoping to offer some historical perspective on flow volumes on the Snowy, extending back to before the construction of Jindabine Dam. And the only gauge extending back enough is the Jaramond River gauge further downstream on the Snowy River, with records back to the early 1920s and up to present. Presented here are the logged scale total monthly discharge in gigaliters for each month, colored by decade. You see that up to the 1950s, in red, orange, and green, on the left of each month, flows would increase from around May and peak around the month of October. Obviously, since the construction of the dam in the 1960s, total flow volumes have dropped considerably, with the marked increase since the return of environmental flows in the bright pink on the right. Here's how the graph looks when total monthly discharge is grouped by period. In red, 1920s to the end of 1950s, in green, the in-between period extending up to 2011, and in red, since the use of environmental flow to present day. When looking at the average daily discharge rates recorded at Dalgety since 1997, you can see the considerable increase since the onset of environmental flows here in blue. Over the past decade, there have been large releases during the months of June and onwards to October, but also in March, as was the case in 2012. To test for any effect of the large spring release on platypus numbers, we surveyed 4 locations between Jindabine Dam and Dalgety, 2 up in the more steep gorge areas marked in red, and 2 lower, marked in yellow, where the land flattens down at the winery and another at Dalgety by the caravan park. We surveyed these sites 4 times, once a month between August and November. In the gorge area, we caught only 4 platypuses in August and September, as depicted on the left panel, while we caught none in the 2 months after the release. This, however, may be a result of higher flow rates decreasing our likelihood of capture. On the right panel, you'll see that we caught more platypuses, with similar numbers before and after the release in Dalgety, and somewhat similar numbers at the winery. As for tracking movements, we glued tags on platypuses. As you can see, I'm using Tennille's hairdryer to hasten drying on some cold nights. In total, we tagged 14 platypuses at the winery in Dalgety, and only one further up just by the dam wall, as a result of low capture rates. In this study, we also wanted to examine more closely fine-scale movements in habitat use, and so we also concentrated our listening stations to detect movements within a single pool. This one is a bit messy, but the figure represents the proportion of detections colored by each receiver for each platypus before and after the release. What the figure tells us is that most individuals maintain the same foraging area before and immediately after the release, with no apparent displacement in location or movements. In conclusion, assessment of past platypus observations indicate declines in both distribution and numbers over the past century, particularly across the Murray-Darling Basin as well as in Queensland, but also highlighted the significant knowledge gaps that must be addressed through monitoring. In our surveys, we recorded large variability in population sizes and demographic structure, with evidence that this was related to dam operations and habitat destruction by clearing of riparian vegetation and destabilizing of riverbanks by livestock. Developing conservation management strategies should be a priority if we are to keep the platypus from becoming endangered in the wild. In terms of spring release, whether the extremely large, in the order of magnitude of 8,000 megaliters per day, restorative spring releases from Jindabyne Dam can drown puggles and barrows is still unknown and likely requires more targeted monitoring. Similarly, we are unsure if the low proportion of juvenile platypuses in some rivers, including the Snowy, is of concern, and again would require further monitoring. To minimize risk of drowning, perhaps the extremely large deliveries should be made no later than early October or perhaps even September. Over the coming months, we will complete analyzing samples we collected, increasing our knowledge on food web structure, individual health, and genetic diversity that will better help us understand and assess the viability of these populations. I'd like to take the opportunity to thank you all, the many volunteers who helped us over the years, Pam and Luke at Parks, landholders for providing access to the river, Snowy Hydro, and Dion at Austral Research and Consulting. Please don't hesitate to contact me if you'd like further information. Thank you for your time.